The aim of the following experiment is to knock down maternal proteins stored in oocytes or overexpress proteins of interest at the point of fertilization of frog eggs. This is achieved by injecting antisense oligos, or mRNA, into enzymatically defolliculated immature oocytes to degrade or overexpress a specific protein. As a second step, oocytes are transferred to progesterone-containing medium, which induces maturation of the oocytes to eggs. Next, sperm is injected into the in vitro matured eggs in order to observe the effect of knockdown or overexpression on embryonic development. Ultimately, the development of oligo-injected oocytes to swimming tadpoles is the functional test of gene knockdown or overexpression.
The main advantage of this technique over existing methods like host transfer is that we can skip the frog surgery process.
Using standard protocols, collect a Xenopus ovary sac containing healthy oocytes in a 9 cm Petri dish containing MBS plus PS. Then, tease apart the ovary into square pieces that are 1 to 2 cm wide. Collect about 5 ml of the torn ovary with oocytes to a new 50 ml tube. Then, rinse the tissues with MBS plus PS a few times. And collect the eggs in 15 ml of fresh MBS plus PS. Now, add 7 units of defolliculation enzyme to the suspension and incubate the suspension with